India's about to get hit with a super cyclone.
Oh boy. Okay. So those aren't very high wind speeds, are they?
Oh no, those are like Cat 5 hurricanes.
Oh, they are, huh?
Yeah.
How big are they?
This one is as big as India.
Oh, shoot.
It's the size of this monster.
Hello, Jesse.
Morning. Nice to be back here. I think I've been here a long time, but
Oh, it's okay. Yeah. We're waiting on a couple more people.
how are you all doing?
I'm doing pretty well. Just starting the week.
Yeah. This is more for the other lab, but I'm gonna finish. Are you just basically just resubmitting your talk for Neuromatch 1 again, or?
I'm actually submitting a different talk, so
Okay.
I'm going to work on that. It's some other, it's a little bit different focus than what my Neuromatch 1 talk was. So
Yeah. I think I'll mostly just have a slightly updated version of the past one and see. I kind of want to ask them about that because I didn't actually present in the moment, but like I don't know if they would discourage me from doing the same thing. There are other things I could present, but I kind of want to. We'll see about that. I guess.
yeah, I don't know. I mean, I guess they'll probably accept anyone. You know, that I don't think there's going to be anything rejected, but they're just going to try to fit everything in. But you know, I mean, I would probably go with what you had before. It's probably easier to kind of develop it and you know, to keep in the same mindset. So I don't know if people are coming here. Okay. Let's see. I have a message. So Ujwal's not going to make it, and able to make it today. So well, let's see. I have I'll just record this, and then we'll kind of go through some things. It might be an abbreviated meeting today, but that's okay. I was waiting for Ujwal and Mayukh were going to give us some updates on their projects. So they're starting community period and of GSOC, and they are going. They're starting out. I mean, it's, you know, I've encouraged them to explore the open worm community, and I think Mayukh is actually doing some work on motion tracking of worms. So he was showing me some of his work, and it looks pretty interesting. He sent me a GIF. I don't have it with me here, but it was just like a basic first pass approximation at it. So we have all this video up on the movement database that basically has a bunch of worms that have been observed in a culture, and they are moving around. And the idea is, you know, you're able to track their movement using different, you know, points of reference, and then, you know, you can say things about the movements, you know, classify them and so forth. So he's actually using a machine learning approach, which is a bit different from what other people have been doing. And so, you know, we'll see where that goes. It looks interesting. There might even be some applications to development in later embryogenesis when you start seeing muscle twitching in the worms, for example. I don't know if we can get video of that. I think there might be some data sets where that might be cleanly captured, but I'd have to look around for that. And anyways, I mean, it looks interesting. So, but their main projects are not starting yet. I had their task board out. I don't know if we're going to get to that. But I just wanted to bring that up. That's what we were going to hear today. But I should ask people if we have any questions or comments, anything we should catch up on here. No? How about you, Jesse? I know you haven't made the meetings in a while, but...
Yeah, I don't have that much to say, but I do want to be involved in DevoWorm stuff. I know I mentioned some things with the Google Summer Code sort of stuff that's already going on, but also I'm not up to date with things like the project list, but I think I'll be finding some things to do on that as well, especially if they can dovetail with other stuff. But I don't know if that's something to really say more about now, but I'll be looking to be involved in that in some form.
Yeah. Yeah, so why don't I go to... Actually, I should go to our task list for the group.
Yeah.
So let me share my screen here. And here it is. All right. You can see my screen.
Yes.
All right. So let me go to the our group board, which is a little bit different from the GSOC specific group board. And then that is projects. And then we have major tasks for 2020. And let's see where we are. So we have a bunch of things that have already been completed since I started the board. A couple of presentations, the Google Summer Code selections, the virtual worm participation. So I sent out a link a couple weeks ago. I know I went over the virtual worm talk, which was well received. And I sent out a link. I don't remember if I sent it in the group email or not, but it's the YouTube archive of that talk. So let's see. I'll probably send the link out again. I don't think I put it in here, but it's archived. It's called Virtual Worm. I think it's Virtual Worm Lectures or something on YouTube, and it's run by someone who's at I think Baylor College of Medicine, and they wanted to give exposure to C. elegans researchers from across different areas of C. elegans research. So there are a lot of good talks there, 30-minute talks. So check that out if you can. Let's see. Oh, Ojwal was also going to present on CC3D. He said he made some progress on the... So the CC3D is the CompuCell 3D project, or the CompuCell 3D program that he's trying to build models in. So let's see what we have in progress. We have this axolotl data set that we talked about several weeks back. And this is the one that is Susan from our group here. She sent me some data. This is the stuff that her and Dick have been working on with the flipping microscope, which is where they take an embryo and they flip it, and they image the sort of this flipping of the. So it's like it's a ball, so it rotates around. We have that data that we're trying to get a handle on. It's we don't have a good path forward on that right now. but we have the data and it's sort of sorted and ready to go. So we need to maybe work on that a little bit. What else? We have some open papers. So these are things that are kind of open, that are on GitHub. So they're papers that are maybe outlines or maybe a little bit more than outlines that either have links from to GitHub or to Google Docs, and you can contribute to those papers. I'm not sure where the link is for that. I think it's in the repository here, the group meetings repository. But I can provide a link to that as well if you're interested. And it's just it's a README file with a bunch of links. I think you saw that at the last meeting.
Yeah, I remember this a while ago.
Yeah. So reason, and you have to request permissions to make edits, but the reason I do it this way is because people want to contribute to something. If there are some ongoing papers or some ongoing ideas, we can just simply someone can request edit access and maybe make some contributions. I mean, it's controlled, so it's not like some random person on the internet gets in there and makes some bad edits or something. But yeah. So I mean, if you're interested in that, we had the student presentations last week, so that if you want to go to the YouTube video of that, it's there. It's just the two student presentations, basically their proposals that they had, or it could have been 2 weeks ago. Well, anyways, what else do we have? And we're still working on the embryo physics archive. That's up on GitHub, and these are lectures that were presented probably about 10 years ago now in that area of time, and there are all these different lectures on embryos and development and physics of development, and so been re-archiving those to a more permanent location and maybe building some lesson plans around that or some curricula around that. But that's something also that you might want to contribute to. And then that's pretty much what we have in progress. We have a couple of things on hold. The DevoZ reorganization, I think that'll be Ujwal's project. The axolotl montaging, which has to do with the axolotl data set. I think that's going to be something we'll probably need to plan out a little bit more in terms of how we want to deal with the axolotl data. I think the idea was, I think I sent this out in an email a couple weeks ago, it's to make this like sort of Google map of the embryo, where you have the surface of the embryo and you can turn it and you can see the surface of where you are on the embryo. And then that's, it's presented in a browser so that you don't have to. There's Susan. So that you can travel across the embryo surface and get a sense of space. Hi, Susan. You're muted, I think. Or you don't have sound. No, I think you still don't have sound. No.
Is the Create DevoWorm ML on Google Classroom part of another project?
I think that's just the stuff that from last fall. So this is this is just the version. Well, I mean, I put that up on Google Classroom so that people could have it all in one place. They could enroll with for it. But it's basically all this. It's basically all the weeks from last fall that we did that, and it goes through different topics, and it's kind of organized weekly by topic, so people can view that, and that's what that is.
Okay.
But I mean, yeah. So this is this was the idea, was to create all of these on Google Classroom, sort of as like a skin for it, and then
Yeah.
So Susan, and you can just type in the chat if you can't get your sound working. Oh yeah. We were talking about the axolotl data and sort of getting a plan together for that. I'm waiting for some people to, oh, you have more? Yeah, actually, that would be good if you could send me more. That would work. We're still working on how to organize it in terms of, how to present it, how to build an interface, and so forth. So, yeah, May 26th. That's fine. I mean, we have enough. I think we have enough data to work on it at some point. But yeah, we're not. It's not like at the top of our queue. But if we find someone who wants to work on it, and we'll probably start planning it out, and it'll be, it'll take a while to see. So, yeah. So that's okay. So I was going through the task board, Susan. So I think you can see my screen here. So I was going through this. So I talked about the axolotl data set, talked about the papers, and then the Google Classroom, which includes the embryo physics archive. And then, let's see. So there are a bunch of ideas that are kind of fuzzy here that we were also talking about the embryo visualization for Docker. So I don't know, Jesse, if you remember that, but that was something that is in association with OpenWorm. So OpenWorm has this Docker container that they have that they want to present all the parts of the OpenWorm project. And we don't have a development part. So the idea would be to build like some sort of model or simulation for that. I know Ujwal is working on simulation. That's what he was going to present on with the CC3D stuff. Like he's got these 3D models that he's going to extrude and use those as sort of. I don't think it's supposed to be like a high resolution model, but basically like something that's just a demo for people. Because we don't have like a lot of the other projects in OpenWorm are dedicated software projects. Like they're doing, it's like a simulation platform or something. So they have like this, they can just put the output of that into it and show people, and they can run that program. And it's actually pretty easy to do if that's what you're working on. So, you have this simulation platform. You just say, okay, build a simulation, and spit out some results, and that's the whole contribution to the Docker file. But this is a little bit different because we don't have like one simulation platform. So, for those of you who aren't familiar with Docker is it's like a sort of like, I don't know if you're familiar with virtual machines, but it's something you can open up on your computer that simulates like a certain set of operating parameters. So the benefit of that is that if you send this Docker file to all different people, with all different types of computer hardware, they can run a simulation or something within that Docker container under the same conditions that you ran it on your computer. So if you're interested in running like a simulation, or you need to run like some other thing that's really dependent on the speed of the computer that you have, or the conditions of your computer, it's actually quite useful. They do use it a lot in reproducibility. So if you want to reproduce an analysis or build a simulation that's reproducible across machines and runs, then you'd use a Docker container. And people have gotten into like distributing software in Docker containers. And the idea is that it makes it easier to install and run without worrying about like versions of your computer and that. Now it turns out that it's really hard sometimes, especially on Windows, to install Docker and get it to run. So there's that.
Yeah.
But it's in theory it's supposed to be a nice way to do this, and that's why they tried, they actually built a Docker container for OpenWorm. It's a early version. So if you have a Mac or a Linux machine, you can run this thing, and it just spits out a lot of images. So it's something that will require a lot of hard drive space. So if you're right up at the top, maybe you don't want to do that. But let's see, we have some questions in the chat. Okay, so that's what Susan was talking about. Okay, she was mentioning about her exam schedule. So yeah, I mean that's what we have. So we have like basically 4 things that are really kind of on sort of on the precipice of getting done. The rest of this stuff it needs a bit more work to really get to the point of something you can just grab onto and start contributing to. Let me see if I can find that link to the papers.
What was the issue number 3 to do complexity measures?
Oh, complexity measures. This is an old thing that I think Dick and I talked about it one time. It was something that is in my DevoWorm folder on my hard drive. So it's really, I think it's okay. Oh, I think it has to do with like measuring complexity in organisms. So we were talking about it in terms of Basilaria when we had a Basilaria discussion. But I don't think we developed it any more than that. Do you remember that, Dick?
No.
Okay. Yeah, I don't think we did too much on it. It was just something we kind of mentioned in passing, and we did, I think, we looked up a couple of papers on it, and I have a folder in my hard drive, and it's like got some things in it. I thought it would be interesting to drive forward, simply because it's kind of the thing we are interested in here, is quantitative.
I had no idea what it was about, and I see that it was made like in February, so I'm sort of a long time ago, but I'm in a reading group about like emergence as the concept, and we recently looked at a few things about layers, or about like one of them was like looking at the concept of levels, and another was looking at like I forgot the last one, but some of the papers we've been looking at might fit with that. So I don't know if I don't really know what was going on with that, but I may have some like adjacent work on that might fit with that. So,
I think it like I know people have looked at looking at biocomplexity and looking at different sort of scales of an organism. So from like cells to genes to the whole hierarchical type of approach. But people put a number on what's the complexity of an organism, right? And the number is pretty dependent on what you choose to measure. So the idea, I guess, would be we maybe would propose some measures that would be good for sort of getting a better understanding of biocomplexity in some way, and something that's robust. I know people have done this in the past, like in different papers, so I think there's a literature, but I think the problem, of course, has always been that it's a pretty it's a pretty abstract thing. Like they have things like scaling laws, which are kind of like where they put a bunch of organisms on a chart. They say this is body size, and it scales with temperature or brain size or something, and you get this curve, and it's a pretty sort of vague measurement. It's like body size, they measure it from like tip to tail, and that's supposed to tell us a lot about this phenomena. I mean, it scales nicely, but what does it really tell us about the phenomena? Because there's it isn't just like this length measurement. The organism, of course, is a 3-dimensional shape, and then it moves around and it behaves. So this idea of even with something like scaling coefficients and scaling laws, those are really based on very general measures that don't really take into account, say, like the entire geometry of the organism. So, I mean, there's stuff like that where it's like you can get into the idea of like, if we could have like a good measure, or we could conceive of a good way to measure biocomplexity in some way like that. We might be able to derive some laws or some sort of general principles about how this unfolds across the tree of life. Because right now, I mean, we have like scaling laws. I think we might have some measures of like, complexity, like with circulatory systems and things like that. People have proposed parameters for that, but I don't know. I don't know what would be appropriate there. It's something to think about. It's probably why we didn't do anything with it, because it's just such a broad area that it's hard to. It would just require thinking about like what you want to measure, and then maybe we can use some real data to measure it. Maybe we can get like simulate something. It just really depends on what we think would be a good way to approach it.
Okay.
Yeah. So let me see if I can find in group meetings this link to the papers. Okay, this is it. This is the README in group meetings. But if you just go to this group meetings link, it'll be like below the repositories. So these are the repositories up here, and then you have these links. We have someone coming in. Mayukh's here. Good.
Okay, I see them. That whole section.
Yeah, so these are different group papers that are ongoing. It's either that it's something I'm leading out, or we're doing something with them. So yeah, just take a look at those links, and then if you're interested, if it's a Google Doc, request access. If it's a GitHub repository, you can, fork it and make a push to the main directory, or, just, even if you don't feel comfortable in GitHub, then you can send some, send some suggestions. Even by email would be fine. Hi, Mayukh. Let's see. Mayukh had a presentation to make, I think, but I wanted to...
Am I audible?
Yeah, you're audible.
Am I audible?
Yes. You're good.
Okay.
I wanted to get an update from you. I wanted to go through one more thing before that, though. And that is the papers. We had so this is the Basilaria paper. This is... Dick, myself, Thomas Harvick, and Ujwal, and Vinay, and Asmit Singh as well, but he doesn't usually come to meetings. But I wanted to make this, I wanted to show everyone this so everyone knows what's going on here. So we submitted the Basilaria manuscript to this book chapter, as a book chapter in this book. And so now it's been accepted for publication. So this was Stanley Collin, he's the editor, and we sent him some additional reviewers, and so we got all that taken care of. So now it's accepted. We have a couple of things to address now. The first is that there are a couple of things that they wanted us to do before we get it published. So the first of these is, in the NN, which is the neural network description section, since the readers might not be familiar with artificial neural networks, it might be good to point out the differences between artificial and natural neural networks. One main criticism is that the backprop algorithm that trains the network doesn't appear to have a biological equivalent. And so that's one thing. I know Ujwal and Asmit wrote that section on the neural network descriptions, and I may have cut out some relevant detail when I was editing it for the first round, but I can look back. I think, though, this might be something that we can answer pretty quickly, maybe with a couple more references. Though there's been a debate about the difference between artificial and natural neural networks recently. And so I'm going to look into that debate. Maybe that's pretty timely to put into the paper. The second one is on the PCA analysis. I don't see any values for the chosen features. So from the diagram, it doesn't seem like there's a lot of variance between the axes. So I'm not sure if this was from what Thomas did or it was in the deep learning analysis, but I'll check. If it's, whatever it is, we'll figure it out. That doesn't seem like it's too hard. And then 3, I hope I'm not missing this, but it wasn't clear how the deep learning network was trained. It doesn't seem to be supervised learning. Is there a diatom or not? It is more given a cleaned up image where it's, okay, I don't know what that means. It seems like it's more of a cleaned up image of where the diatom bodies are. Could use more explanation. Okay. So that's going to be, so the way we did this, we had the pre-trained model, and we actually moved that detail to a supplemental file on GitHub. So that might be something we'll have to kind of explain more in the paper. And then there are a couple more citations I want to add in that are, sort of in the last year or so that have come out. There have been a couple of reviews on deep learning and biology, so those we should add. And then I think maybe a couple more references on some of those points I mentioned. So congratulations, everyone, we got that paper accepted. And if you have any notes on that, I think I forwarded an email with the acceptance to everyone who was involved. If you have any requests for any of these changes that we need to make, let me know. Let me I'll put the link to this set of action items in the chat here. And so if you need to request permissions to that, do so. But this is basically our action item list. So we're going to try to get this done the next couple weeks, I think. We'll just have to follow up on these things and add in a couple of new references. So it should all be pretty easy. I think it'll be pretty easy to do. But they were pretty impressed with it. And I think, yeah, it's a good paper, definitely. So then that's what will be in this book chapter. And then we have another paper, of course, on the basal area behavior. And so that's this paper here, the basal area site. Well, I have this paper on collective pattern generators, which is the basal area based paper, and then the basal area psychophysics. So we have two sort of outlines, and then these are in the Google Doc. Both are in Google Doc. One is in GitHub. So we can look those over, and if we have any ideas about how to proceed on that, I should actually present on some of that to give people an idea of what's going on, or what I'm thinking and what other people are thinking on that. So that should be that's something else we can follow up on, I think, because I think we'll have some momentum on the basal area data. But Mayukh, I guess I want to hear from your side. I want to hear an update on what you've been doing this week.
Hello? I couldn't hear that clearly. Could you just repeat it again?
Oh, could you give an update on what
really
you've been
bad.
doing? What was that?
The network's really bad.
Yeah. So you can type into the chat too if you want to update us that way. Yeah. Susan, if you say something, you can see. No, I can't really hear you. Still not, yeah. So, yeah, I don't. I don't know if Mayukh is giving us an update. Yeah, I think he's gonna shut his camera off. That's probably a good idea. Okay. Operator theory and math. I don't. Yeah, give an update on what you've done in the last week. So you sent me the that thing on the movement of the worm. So you were actually doing some movement tracking.
Yeah. Yeah, I can just share my screen and show you guys right now.
Okay.
I can just explain what's going on in the node. Would that? Yeah, fine.
Yeah.
Entire screen. I'll just. Hold on.
Okay.
Can you guys see my screen?
Yeah, it's coming up.
Huh?
There we go. You can see it. Yeah.
Okay, fine. Let me just.
Yeah, you made a push this week, Friday, I think. So that's, yeah, it's Jupyter Notebook.
I think I made the push. I'll show what's going on in the notebook because
Yeah.
that will be important, I guess, later on.
Yeah.
I'm trying to do something on the movement database that you guys have.
Okay.
Movement database, it's like really big. There's a lot of stuff there
Okay.
which I can work with. And I've been reading a lot of papers on locomotion. So so I'll just go straight into the details. So initially, I was trying to track, the first thing that I want to do is there were a few papers which I read recently. And those papers, they talk about how locomotion can be used to detect certain kinds, the certain subtypes of the worms, from N2 is the white type, I guess, N2 from the other mutants. So the time series data of the worm's locomotion can be used to classify the worms. So but the first that would be to collect the right data. So the data would be like any kind of video, which would be later on we can just use a video file from that. The model could track the worms and then classify them. So we just in this pipeline, there's a frame taken, then we threshold, and after thresholding we find the rectangles. These are the key. This rectangle is again, there's a certain amount of padding that I've given to the rectangles to make sure that all of the worm is within the frame, because the rectangles were not completely accurate, so I had to add some amount of padding. So moving on, then I just segmented the image. What I did was, I used an operation called bitwise and within OpenCV to segment out the worm and eliminate the background. And then I could save all of these images from a very long video file, thousands and thousands of segmented worm images into a folder. And that pipeline is basically ready. But then the latest update on top of this would be on this notebook. Can you guys see this notebook?
Yeah.
So the next objective that I was thinking of was to find the head and tail of the worm, because that would be important, right?
Yeah.
Hello?
Yes. Yeah.
Am I audible?
Yes.
Yeah, so the next step was to find the head and tail of the worm. So that is exactly what I did. Like, as you can see here, I found out the head and tail of the worm using some more algorithms in OpenCV. So then I want to make something like there's a lot of data on that movement database, right? I want to use this kind of a model. This is not even a model. This is like a pipeline which extracts metadata from the images and the video of the worms, right? So I'll be using this kind of and training a model which would try to classify those worms from the movement database that's there.
Okay.
For now at least. I'll be working on this pretty soon.
Yeah. That looks pretty good.
Yeah. That's
So
it.
just, yeah, you'll have. Well, yeah, I was looking over the pull requests that you made, and it looks sometimes the Jupyter notebooks don't render in GitHub for some reason. I don't know why, but we can always just download it or go to the NV viewer. I mean, that's something that can be done. It's not.
Yeah, MDView. I'll just put an MDView link on the email. I'll put the MDView link.
Yeah. So I mean,
forgot to do
overall,
it. I'll be sending on the email.
Yeah, please. The README is a good place to put a lot of stuff. So we'll discuss code annotation.
Yeah, we'll have to.
But yeah, I like to have everyone.
We'll have to do a lot of stuff later on.
Yeah. But yeah, this is, I think, a very good start. I think we have, it's a good, well, first of all, it's a good practice run for your project. But also, I think there's probably something that can be done with just general motion data. I mean, there's probably some, like muscle twitch data in the embryo. But more generally, there's a lot of, like, microscopy of movement of C. elegans. And, they have, like, the state-of-the-art in motion tracking is where they'll take, like, a proprietary algorithm, and it's largely for, like, people when they're doing microscopy to track and align images. But I think a lot of motion data is also useful for kind of getting a handle on the higher order details of movement. So, I mean, that's what they've been doing in the movement group in OpenWorm, except they haven't used machine learning. They've been using mathematical modeling. I'll send a recent video from one of the people who are Andre Brown, who works in OpenWorm on the movement side. And they have this thing called Terpsy Tracker, and that's their sort of state-of-the-art. And he gave a presentation.
Yeah, I'll see that.
Okay, good. He gave a presentation on it.
So the data.
Yeah.
Most of the data on the movement database is from that.
Okay, yeah.
Almost all the data on the movement database is sourced from the Terpsy tracker.
Okay, yeah. So that's another good thing to like kind of get.
My algorithm is pretty similar, but the thing about this is that we can integrate this one into Ujjwal's web backend.
Okay.
This is more portable.
Yeah.
And this can be easily fused in with the deepest unit. That's the objective in the end. Terpsy tracker is like a complete product.
Yeah.
But mine is like a portable prototype which can be plugged into a deep learning model so that it can be scaled later on. Or it can be used as a Python library as well. By the end, I could just make it all into a Python library, which can be used to track and do anything, all sorts of stuff on the worms.
Yeah. Yeah, I think having a more general.
Because that's one of the objectives.
Yeah.
OpenWorm already has some kind of tools I've seen on GitHub. Like, we can import a small library that OpenWorm has, but that library seemed a bit obscure for my purpose, so I didn't really use it.
Yeah, that's fine. I think it's good to have something.
There's a lot of stuff on it.
Yeah.
And like, whenever I'm searching for data online for the C. elegans worm, I'm always ending up on the movement database. Because that is like the biggest place.
Yeah.
So I have to start there.
Okay, yeah.
That's almost all the papers that I've read, all the data is sourced from there. So what do you think? Like would the classification model that I just talked about, like if you classifying between N2 and like, there are 5 basic categories, right? I don't remember. Like N2 and then 4 else, I guess. Like 4 mutants and then the N2. Yeah. So there are 5 categories. So would it be useful if I make a model which classifies between these 5 categories based on the movement data?
Yeah.
It is possible as far as I read on the paper.
Yeah, I think
It is
that's
possible.
yeah. I think you should be able to like get some sort of difference there. I'm not familiar with
There is
the
some
way
sort of difference in the movement data.
what was that?
Most of the time went into paper reading. What?
Yeah, I couldn't hear you.
Yeah, most of my time last week went into paper reading. I couldn't really code much.
Yeah, okay. Well, no, that's okay. Yeah, I think the community period is for like getting familiar with things that are going on in the
Because I'm really very familiar with worms. Last time I studied biology was like 4 years back. So you can guess.
yeah.
So I'm really learning a lot about this worm more than anything.
Yeah, I saw your blog
Yeah.
post. So the blog post you said that you were doing some reading, which is good. If you need any other references, I can, let me know. I do encourage you to read a lot on different things that you need to get familiar with. Because I think that's important to know, instead of diving right into something and not knowing what you're analyzing, it is pretty good. It's a pretty good idea to read about it. Make sure that you're.
Ujjwal couldn't attend today, right? Ujjwal. He didn't come.
What was that?
Ujjwal.
He couldn't make it today.
Yeah.
So
Got some problem, I guess. He told you, right?
What did he do?
Did he notify you? He's got.
Oh yeah,
Like he won't be able to come to.
yeah, he notified me in advance.
He told.
Yeah.
Yeah.
So if you can't make the
So I
meeting,
think I'll be making another push by tomorrow after I'm done with tracking the head and tail of the worm.
okay.
So I've been trying to do that.
Yeah.
I'll make another push tomorrow. Maybe. Depends.
Okay. Yeah.
And Ujjwal will be making a website, right? Will you also overall the main DevoWorm website?
Was that about the main DevoWorm website? Or?
The DevoWorm homepage, like the green one.
Yeah.
You'll be overhauling that as well.
Yeah, that's
A lot of
I'm going to
websites.
be working on that. Yeah.
Yeah. And what would Krishna Katyal be doing? Another guy who was there last meeting.
Yeah, he might. I don't know what he'll be doing yet. We'll have to talk about that some more. We haven't talked
So,
about it.
because I saw the issues and I saw that you wanted to assign something to Krishna Katyal.
Well, yeah, I think I sent you an invite for like as a collaborator. I don't know if you got that in your email, but if you can, if you can accept that, and then if you become a collaborator, I can assign people tasks like by their ID. So that might be good too over the course of the summer.
Okay, wait, let me check.
Okay. So
if I get the right data, the model that I'm talking about, this classification model
yeah,
that I'm talking about, if I get the right data, I might just be done before phase 1.
yeah,
Yeah, so after that I'm planning of like, till then I think I'll be a little more familiar about the worm, so I'll go with another model.
okay,
There's multiple models, why not? Different purposes.
yeah,
Yeah, it's going to be exponential in a way because when I know what's going on, when it is only then can you understand what's going on in the data itself.
Right.
Because I have to understand data really well first before making any decisions on a model.
Right. Yeah, I think that's a good
Yeah,
idea.
so that's pretty much. Even Vinay Roma couldn't come today, right? Vinay.
Right. Yeah.
Oh. Yeah.
So yeah, if you have any other, like during the week, if you have questions, you can send them to me on Slack. I encourage that. Usually we can work things out if you have problems with technical issues. Also, if we have, and I make this as a general announcement here, we do have a Friday meeting that I do. It's at 4 p.m. UTC, which is noon Eastern Time, 11 Central Time in the U.S. But we do this meeting on Fridays. It's more informal than this. It's just if people want to stop by and discuss things. I have that space open as well. I'll send a link out in the email about that. I used to do this last summer, I think, and then I stopped doing it for a while. And now I think it's time to revitalize it a bit. Just, especially for like Google Summer of Code people, if they want to talk about issues that they have, it's a good time to do it. But that's separate from this meeting. That would just be like a place to like maybe debug some code or discuss some ideas, show some screen, share your screen for some things that you might want to discuss. So, and then that I extend that invitation to everyone because we have so many things going on in the group that if you want to catch up on things, we have another time to do it. So, well, thank you, Mayukh, for your update. Like I said, Ujwal couldn't be here. Yeah, and we'll be doing these updates every week. And then like the coding period starts in about 3 weeks. Also, I wanted to mention, and Ujwal's not here, but we have this this Wednesday. We have an event in Slack, and so this is the one where we have the
office hours.
Yeah, office hours, and so I'll be there, and I want you to join me in this channel, office hours, and we're going to just kind of talk about like typing it out. We're going to talk about the project, sort of give a little introduction, maybe put some screenshots in. And, it'll be there for people in OpenWorm so they can see, like, it'll be like, archived in that channel so people can go back and look at it. So it'll be a nice way for them to get, like, some more information about your projects. And so OpenWorm... Yeah, office hours will be Wednesday at 3 p.m. UTC, which is 10 o'clock Central Time and 11 o'clock Eastern Time in the U.S. And so that'll be happening as well. So we have a couple minutes left. I was gonna, I had a backup where I was gonna present some papers. So and I've been waiting on this for a couple of weeks. So what I'll do is I'll just kind of like show people these papers and give a link to the repository where I have them. I have them in a Google Drive as usual. And so I've been trying to do this over, these meetings just to bring people's attention to papers that have been published. And in this week's, wait a minute. Yeah. So this is actually two papers. It's in developmental biology. One is a paper on modeling stripe formation in zebrafish tail fins. And so this is a new paper out on the archive. I don't know the details as to where it'll be ultimately published, but it's looks pretty interesting to me. It's in collaboration with people at Facebook and Citizens Bank, I guess. It's data science and it's development. So it's kind of what we talk about a lot here. So, so the abstract is As zebrafish develop, black and gold stripes form across their skin due to the interactions of brightly colored pigment cells. These characteristic patterns emerge on the growing fish body, as well as on the anal and caudal fins, which are at the tail end of the fish. While wild-type stripes form parallel to a horizontal marker on the body, patterns of the tail fin gradually extend distally outward. Interestingly, several mutations lead to altered body patterns without affecting fin stripes. Through an exploratory modeling approach, our goal is to help better understand these differences between body and fin patterns. By adapting a prior agent-based model of cell interactions on the fish body, we present an in silico study of stripe development on tail fins. Our main result is a demonstration that the two cell types can produce stripes on the caudal fin. We highlight several ways that bone rays, growth, and the body fin interface may be involved in patterning. So they have this, they present this model. This is the zebrafish, and this is just kind of like where on the fish they're looking. They classify the cells on the surface of the fish. You have melanophores, xanthophores, then xanthophores. These are just different types of cells that are doing different things, contributing to the striping pattern. And then you have a different pattern on this fish, and you have the same set of smaller set of cells, and this is their sort of organization in space. And so then they built this model. They say it's an agent-based model, so I thought Jesse might be interested in reading a bit about this because he's interested in agent-based modeling. Then they propose some multiple mechanisms, and then their model. And again, they just use their different cell types. They look at the position of the center of the fifth melanophore at CT. So they look at each cell in each class at different times. So they have these functions for each cell class and the different cells in that class. So the class is indexed. And then they run the simulation. And then they propose some so they put some parameters into the model, and then they make some predictions. And so it's a pretty long paper. So this is one paper that's one of the ones I wanted to show. And then in the last 2 minutes, I'm going to show this paper just to let you know it exists. Cell-cell interaction and the diversity of emergent behaviors. And this is a review in IET systems biology. So this is it's more of a review paper. And so they introduce multi-random Boolean networks, which is a model for looking at interaction among cells. And so we talked about cell-cell models before, but not too much. I mean, cellular communication is what they focus on here. And these are Boolean models. So it's actually a nice, I think, contribution to that area. They propose some mechanisms, and then they analyze their models. So that's another paper. Yeah, they actually use RBNs. Yeah, they use random Boolean networks. So yeah, it's a long paper. I don't want to get into it again, but I wanted to make it aware that it existed. So that's another paper. That's kind of the state of emerging state of the art of the field. So what do we have here? We have a comment by Susan. Vibration when the embryo is developing can change the pigmentation pattern. Yeah, that's an interesting observation. There are a lot of different ways. People will propose different models for how striping happens. You have your classic model, like your Turing model, reaction diffusion model, but there are other things that contribute to it as well. And I think, maybe one thing we can do is give a presentation on, at some point someone can do this if they're interested, give a presentation on some of these mechanisms. And kind of get into this. Yeah, send me the reference for the vibration work. That'd be great. We can maybe present it here, or we can at least make people aware that it's available. But yeah, send me the reference. And but yeah, I think we should follow up on that as well if people are interested. Definitely a lot to explore in that area as well as like machine learning. And then we talk a lot about machine learning, but agent-based modeling, and I think a lot of these cell models are interesting in their own right because they give us, a good, like we're talking about measurement, complexity measures. One way to do that is to have, a good model of sort of emergence, coming from like, say, a bunch of cells. How do they form a pattern? Or, even like how gene expression contributes to cells and their identity and their patterns. So those are all things that are, interesting. So yeah, again, we'll be meeting next week. We're at the top of the hour, so we'll be meeting next week again at this time. We'll be getting updates from Mayukh and Nicole, and we'll try to do like 10-minute updates. Okay, Mayukh, just be prepared for like a 10-minute synopsis of your work, and then we'll discuss it a little bit as well. But then we also want to give time for people to give maybe a presentation on something or whatever you want to do. So if you have anything, let me know. Contact me on Slack if you have any good, interesting ideas or something. Let me know. So thanks for meeting, everyone.
Okay.
Okay.
Bye.
Take care. Bye.
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 Take care.
All right. Take care.